Hi students, welcome to E-Pathashala. I am Dr. Kalpakam Polasa, former Director in Charge, National Institute of Nutrition at Hyderabad. In today's module, you are going to learn about the regulation of glucose in the body. What does the term hyperglycemia, hypoglycemia mean? How the glucose is maintained in the body within a very narrow range, its importance and the consequences if it is not maintained within the desirable levels, the control of blood glucose, the role of sympathetic parasympathetic nervous system, and then finally the drugs that are used in the management of diabetes mellitus. Fasting and postprandial blood glucose. The glucose metabolism in the body is regulated by insulin, which is a hormone produced by pancreas. The level of glucose in the morning is referred to as fasting blood sugar and is at its lowest level. A non-diabetic adult has fasting value of 80 to 110 and after meals, that is postprandial, has 120 to 140 milligram per 100 ml. If the fasting value of blood sugar is 110 to 130 milligram for 100 ml and if it increases to more than 140 for 100 ml 2 hours after food, then the person may be pre-diabetic or has diabetes. Impaired glucose tolerance or IGT. The individuals with IGT have blood glucose levels that are higher than normal but not very high as a diabetic. This condition is diagnosed by conducting a test known as glucose tolerance test. 75 grams of glucose is given to an individual who is in fasting state for 8 to 12 hours. After 2 hours of drinking the glucose, blood glucose is estimated. In normal adults, value is below 140 milligram for 100 ml. In individuals with impaired glucose tolerance or IGT, the blood glucose values range between 140 and 199 milligram for 100 ml. In diabetics, the two are blood glucose after 75 gram of glucose administration, it is 200 milligram for 100 ml or even above that. The persons with IGT have the risk of developing diabetes and it is therefore also known as pre-diabetic condition. With diet modification, exercise and other lifestyle changes, it may be possible to delay the onset of diabetes. However, most of the pre-diabetics become diabetic sooner or later. Let us see now what are the terms hyperglycemia and hypoglycemia. When the blood glucose levels are higher, it is referred to as hyperglycemia. If the blood glucose values are lower than 70 milligram for 100 ml, it's termed as hypoglycemia. In diabetes mellitus, there may be persistent hyperglycemia due to poor control of blood sugar. It can also fall to very low levels with lack of adequate food intake, inappropriate medication, or strenuous exercise on fasting state. The level of blood glucose is also expressed as molar concentration, that is as millimole per liter abbreviated as mm by L or as milligram per deciliter. The molecular weight of glucose is 180. The difference between the two units is by factor of 18. So 1 millimole for liter is equal to 18 milligram per deciliter. Regulation of blood glucose. Regulation of blood glucose means that the blood glucose level should be maintained at constant value without fluctuations between high and low levels. The levels maintained depend on two hormones, that is insulin and glucagon. When high amounts of glucose are present in the blood, insulin, which is produced by the beta cells of the pancreas, is released. When blood glucose levels become low, glucagon, which is produced by the alpha cells of pancreas, is released. There are other hormones too which influence glucose regulation by controlling the sympathetic nervous system. Regulation of blood glucose is very important for normal physiological process and to be alive. If glucose is not provided to brain, it could result in coma and eventually in death, as brain cannot store glucose. Hormones involved in regulation. Insulin hormone, which is the key to blood glucose regulation, is released from the beta cells in the islets of Langerhans present in the pancreas when the level of blood glucose is high. When glucose enters into cell, adenosine triphosphate is produced by mitochondria in the Krebs cycle and electron transport chain. The increase in the concentration of ATP in the cells closes the channels, preventing the entry of potassium cation to be transported into this cell. This results in opening of the calcium channels and facilitating the inflow of calcium cations. The calcium cations are important in the vesicle excretion. There are proteins called as the snare proteins. Vesicles or V-snares, which are incorporated into the membranes of transport vesicles during budding, and target or T-snares, which are associated with nerve terminal membranes. V-snares get entangled with the T-snare protein on the beta cell surface. Evidence suggests that T-snares form stable subcomplexes serving as guide for V-snare binding to complete the formation of the snare complex. When the calcium ions bind with snare protein, the vesicle merges with the cell membrane, squeezing out insulin, which is excreted in blood. Transportation and metabolism. Insulin travels through blood to muscle, brain, adipose tissue and other organs. It binds to a dimeric transmembrane receptor. The receptor autophosphorylates and causes severe downstream pathways leading to glucose regulation, energy storage and DNA transcription. In the glucose regulation pathway, the receptor autophosphorylates, resulting in phosphorylation of insulin receptor substrate 1 IRS1, which in turn phosphorylates phosphatidylinositol 3 kinase, which then cleaves the N-phosphatidylinositol 4,5 bisphosphate. This leaves diacylglycerol in the cell membrane and inositol 1,4,5 triphosphate in the cytosol. The IP3 then travels to smooth endoplasmic reticulum (SER) and cause calcium channels to open in the smooth endoplasmic reticulum, releasing cations into the cytosol. The DAG activates kinase called as a protein kinase C, which also opens calcium channels in the SER. This helps in binding of vesicles to cell membrane. These vesicles have GLUT4 proteins embedded in the membrane. The GLUT4 proteins are channel proteins that allow glucose into the cell. After the vesicles bind with the cell membrane, glucose flows through the GLUT4 protein and into the cell, reducing blood glucose levels. Major metabolism of glucose occurs in the liver. If liver is damaged due to any reason, the glucose regulation is affected. Kidney is another organ involved in breakdown of insulin. Glucagon. When the level of glucose is low, glucagon from pancreatic alpha cells is released into the blood. This results in inhibition of glucose metabolism and glycolysis. It activates the formation of glucose by converting stored glycogen in liver and stimulates gluconeogenesis and synthesis of glucose from pyruvate, lactate, glycerol, glucogenic amino acid to pour glucose into muscle tissue. Glucagon is released from the alpha cells of the islets of Langerhans in the pancreas. Glucose enters the cells through SLCA1 channel proteins and is then used to produce the ATP. When ATP pool increases, it opens the potassium channels. In the beta cells, the reverse happens. When the ATP pool increases, the potassium channels are closed. In the alpha cells, when the intracellular concentration of potassium increases, the calcium channels open up, facilitating the binding of vesicles containing glucagon. This results in release of glucagon into the blood. After the glucose reaches the liver through blood, it binds to a transmembrane protein called G protein coupled receptor. The membrane associated G protein is phosphorylated with GTP, and the alpha unit of the G protein activates adenylate cyclase. Adenylate cyclase converts ATP to cyclic AMP, cAMP, which then activates the protein kinase A. Protein kinase A phosphorylates the phosphofructokinase and fructose bisphosphatase 2. Subsequent to phosphorylation, the activity of PFK2 decreases, and this results in the decrease in the production of fructose 2,6 bisphosphate F2,6BP. High concentration of F2,6BP induces glycolysis or the breakdown of glucose. Therefore, the decrease in the activity of PFK2 reduces the levels of F2 and 6BP. Further, FBPase2 gets activated, which helps in breaking down F2 6BP, facilitating in lowering the levels of F2 6BP. Low levels of F2 6BP causes the activation of FBPase1, which increases the gluconeogenesis. The PFK1, which is involved in glycolysis, is not activated. Overall, these two activities result in increase of blood glucose levels. The autonomous nervous system, which includes the sympathetic and parasympathetic nervous system, also play a role in glucose regulation. During extreme stimuli, the sympathetic nervous system is set into action, known as fight or flight response. The brain sends signal to increase the heart rate, cause bronchial dilation, and hepatic glucose release. The brain also sends signals to the adrenal glands. Epinephrine is released from the adrenal glands and is circulated to all parts of the body by blood. In the liver, it stimulates the release of glucose and thus increases the level of glucose. Epinephrine binds to a transmembrane receptor binding site known as the beta-adrenergic receptor protein in liver. The receptor activates a G protein, which then activates the GFP to GTP, and the activated G protein diffuses via the membrane. The G protein then binds with adenyl cyclase and activates it. AC converts ATP to cAMP, which binds to protein kinase A, which then phosphorylates specific protein. The phosphorylase is phosphorylated and it converts glucagon to glucose 1-phosphate, which is converted to glucose 6-phosphate by phosphoglucomutase. The G6P is then used for energy requirements during sympathetic nervous system responses, which increases the ATP pool in the muscle and brain. This hormone pathway gets reversed when the parasympathetic nervous system operates. The parasympathetic pathway is termed as the rest and digest pathway, which is opposite to the fight or flight response produced by the sympathetic nervous system. The rest and digest pathway helps in inducing a state of equilibrium in the body. Both the two systems are part of the autonomous nervous system, which is responsible for involuntary and reflexive functions of the body. What happens to insulin in diabetes? There are many types of diabetes. The type 1 diabetes, also known as the juvenile diabetes or the early onset diabetes, is discovered in early life, and the people suffering from type 1 DM do not produce or produce too little insulin. This may be due to the damage to the pancreatic cells, and it may be genetic or may be due to autoimmune disorder. These individuals have to be administered daily insulin injection. In the type 2 diabetes mellitus, the body tissue is resistant to insulin uptake or insufficient insulin is present. These people are advised to modify diet, exercise, and they are given oral drugs. There is also a condition known as gestational diabetes which develops during pregnancy. These women have to be managed by insulin. The role of insulin and glucagon in blood glucose homeostasis. The control of blood glucose should be within a narrow range of 4 to 5 millimole per liter, which is maintained by action of hormones, insulin, and the glucagon secreted by pancreas and epinephrine secreted by adrenal glands in the target organs. The balance between the activities of these two enzymes determines the predisposition of an individual to diabetes. After consumption of a meal and the process of digestion, insulin is secreted for metabolizing the glucose, and this process helps in lowering blood glucose levels. When the level of blood glucose is low, glucagon is secreted. This hormone has effects on many other tissues, notably the liver. Glucagon releases stored glycogen from the liver for conversion to glucose and restore desired blood glucose. Glucose also induces the liver and muscle cells to synthesize glucose from other nutrients like amino acids and fats. Glycogenesis. It refers to synthesis of glycogen which is stored in liver. The synthesis takes place during the rest cycle after the Cori cycle in the liver. This process is activated by insulin level due to high blood glucose. Glucose is converted to glucose 6-phosphate. The enzyme mutase converts G6P to glucose 1-phosphate, which then converted by the enzyme UDP glucose pyrophosphorylase to UDP glucose. The product pyrophosphate is hydrolyzed into 2-phosphate molecules by the enzyme pyrophosphatase. 1-glycogenin is a homodimer molecule that has tyrosine and aromatic amino acid on each of its dimer. The amino acid moiety functions as anchor for reducing end of glycogen. The glycogenin adds 8 UDP glucose molecules to each tyrosine. The enzyme glycogen synthase binds to growing glycogen chain. All the while, the addition of UDP glucose to glycosyl residue on the non-reducing end of glycogen takes place. Glycogen branching enzyme glycosyl 1,6 transferase makes branches and catalyzes the transfer of an oligosaccharide fragment to another similar fragment. Cori cycle. This cycle is also known as the lactic acid cycle. It's named Cori cycle after its discoverers Carl Ferdinand Cori and Gerty Fori. In this metabolic pathway, lactate is produced due to the anaerobic glycolysis in the muscles, which is transported to liver to produce the glucose. If glucose returns back to the muscle, it is metabolized to lactate again. The muscle activity needs ATP that is provided by the metabolism of glycogen in skeletal muscles. This breakdown process is known as glycogenolysis, as it releases the glucose as G1P, which is converted to G6P by enzyme phosphoglucomutase. The G6P then enters glycolysis or can go into the pentose phosphate pathway, which provides ATP to muscle cells as source of energy. In the presence of oxygen, the pyruvate can be source of ATP. In the absence of oxygen, pyruvate is converted to lactate that enters into lactic acid or Cori cycle. This cycle is significant as it prevents metabolic acidosis or lactic acidosis. It can result in serious medical condition including death. However, in normal condition it is transported out of muscle into the liver. Excess of lactate is excreted from the kidney, but in people suffering from renal diseases this may be impaired. Let's see now the mechanism of action of drugs which are used in control of blood glucose. For type 1 DM, insulin injection is administered. For type 2 DM, diet and exercise are tried first to control the blood glucose. If this is not effective, oral drugs are used to control the blood glucose. Drugs used in the management of diabetes are based on the biochemical mechanisms involved in blood glucose regulation, reduction of insulin resistance, metformin and glitazones. These drugs decrease the quantity of glucose produced by liver and enhance the sensitivity of muscle cells to insulin. Metformin inhibits glucose produced by the liver and also reduces the absorption of glucose. It improves the function of beta cells in the pancreas. Pioglitazones reduce the glucose synthesis in the liver and improves the sensitivity of liver, fat and muscle cells to insulin. Stimulation of insulin release by sulfonylureas and meglitinides. Sulfonylureas and meglitinides stimulates the beta cells in the pancreas to release more insulin. These act very fast but have shorter effect than the sulfonylureas. They are ingested to increase insulin production during meals, slowing the absorption of sugar from the gut, acarbose. The carbohydrates in the food is metabolized to simple sugar before absorption by the enzyme called alpha glucosidase present in the gut. Acarbose block the activity of this enzyme, delaying the absorption of sugar from gut. This prevents the steep rise in blood glucose levels. DPP-4 inhibition, gliptins. Hormones known as incretins present in the bowel stimulate insulin release from the pancreas after consumption of food. These hormones include glucagon-like peptide, which is broken down by an enzyme called dipeptidyl peptidase 4. The DPP-4 inhibitors are called gliptins, that is, examples are the sitagliptin, linagliptin, saxagliptin and vildagliptin. These drugs prevent the breakdown of GLP-1 so that more insulin is produced in response to food consumption. This reduces the postprandial hyperglycemia. The gliptins also inhibit the release of another hormone from pancreas called glucagon, which acts against insulin. These two functions of gliptins help in improving the blood glucose control. Increased excretion of glucose in urine. Gliflozins lowers blood sugar by increasing its excretion in the urine. Normally, the kidneys filters glucose out of the blood and is reabsorbed back into the blood by a mechanism called sodium glucose cotransporter 2. The kidneys normally reabsorb glucose back into the blood even when the levels of glucose in the blood are too high, as in diabetics. Gliflozins act by inhibiting the SGLT2 transporter. This allows excess glucose to be filtered out of the blood and excreted in the urine. Diet modulation of blood glucose, glycemic index and the glycemic load. Diet should be low in glycemic index, that is, capacity of food to increase the blood sugar, and foods that increase the blood sugar are known as high glycemic index foods. The quantity consumed is also very important. Hence, glycemic index and quantity consumed is as important, and taking into account both these two factors, glycemic load is calculated. Ultimately, the elevation of blood sugar level after consuming meal is dependent on both the glycemic index and the portion size or the glycemic load. High fiber containing diet has low glycemic index, and fiber reduces the insulin resistance and gives satiety, restricts food intake. It reduces elevation of postprandial glucose and thus has beneficial effects on glucose metabolism. In addition, soluble fibers slow the gastric emptying time and slows the glucose absorption and has beneficial effect to the diabetes. There are many cereal products fortified with dietary fiber, and these are helpful in controlling the increases in the plasma glucose levels. You have learnt how the blood control is achieved in the body through the homeostasis involving lot of hormones, the parasympathetic nervous system, the sympathetic nervous system, the role of insulin, and how diet and physical activity modulations can go a long way in controlling and management of the diabetes. You have also learnt the different categories of drugs that are used in the treatment of diabetes mellitus, and why different people are given different type of drugs, and the management of diabetes is achieved only by being physically active, by a judicious combination of diet and medicines.